Hi, this video presents a brief molecular dynamics simulation of a ternary complex involving EGFR, the molecular glue CFT8919, and an E3 ubiquitin ligase. The simulation provides atomic level insight into how CFT stabilizes the interaction between EGFR and the E3 ligase, a mechanism that underlies targeted protein degradation. Molecular dynamics simulations can be used to provide critical insight into how proteins and small molecules move and interact at the atomic level over time. These simulations help visualize the flexibility, stability, and transient interactions within biomolecular complexes that cannot be captured by static crystal structures alone. To construct the molecular ensemble of Cereblon, CFT, and EGFR, I began by retrieving high-resolution crystallographic structures of the Cereblon E3 ligase and the EGFR from the protein data bank. And here is the EGFR. Using a variety of molecular modeling software, I positioned the two proteins in proximity such that their respective binding interfaces could potentially be bridged by the molecular glue. Next, I generated a putative structure of CFT8919 based on information available in patent literature. While it may not exactly match the molecule currently used in clinical trials, it represents a reasonable and chemically plausible approximation for modeling purposes. CFT is a molecular glue composed of a pomalidomide-like moiety, which binds to cereblon, linked to a ligand that targets the protein of interest, in this case EGFR. The specialized linker bridges these two components, anchoring one end of the cereblon binding pocket and positioning the other end near the allosteric binding site of EGFR to promote formation of a stable ternary complex. Here is CFT being positioned in the cereblon binding pocket, and the other end of CFT near the allosteric binding site of EGFR. Thus, CFT was arranged in a structurally plausible conformation, guided by known ligand orientations within their respective binding pockets to promote the formation of a stable ternary complex. Additionally, a well-known ligand was placed in the orthosteric site of EGFR to reflect its known binding mode. Next, topology files and necessary custom force field parameters were generated for the full complex using a suite of tools, including VMD, NAMD, Collab, OpenMM, AlphaFold, Python, SwissParam, and LabPsycho's artificial intelligence. Finally, the ensemble was solvated and embedded in a periodic simulation box in preparation for molecular dynamics simulation. And here is an outline of the periodic box. Finally, the ensemble was solvated and embedded in a periodic simulation box in preparation for molecular dynamics simulation. In this video within a video, we present an MD simulation of the ternary complex formed by Cereblon, the molecular glue CFT, and EGF-Receptor. By examining this simulation, we gain valuable atomic level understanding of how CFT bridges Cereblon and EGFR, insights that can guide the rational design of improved therapeutics. Such simulations are a powerful tool in modern drug discovery, helping accelerate the development of more effective and selective treatments. By analyzing the results of the simulation, we can identify specific atomic level interactions between the drug CFT and key residues on both Cereblon and EGFR. For example, hydrogen bonding, pi stacking, and hydrophobic contacts observed during the simulation help pinpoint which parts of the molecule are essential for stable ternary complex formation. These insights can be used to guide the rational design of next-generation CFT analogs with enhanced binding affinity and selectivity, improving therapeutic performance and minimizing off-target effects. If I zoom in even closer, we can see bond distances between key residues. I am zooming in on the molecular dynamics simulation to highlight key EGFR residues located within 2 to 3 angstroms of CFT, revealing a close atomic level interactions that may contribute to binding stability. I also emphasize the clinically significant L858R mutation site, allowing visualization of its spatial relationship to the molecular glue within the ternary complex. In summary, this ensemble and molecular dynamics simulation offers atomic level insight into how molecular glues like CFT may stabilize EGFR Cereblon interactions, providing clues to their mechanism of action. Furthermore, these studies can also help guide the design of next generation protein degraders.